Today we have our first webinar in the BioGlue Center, and it is a great moment to give the floor to Peter Herder. I could not think anyone better or more appropriate to start this webinar series for the center. I can tell a few words and introduce him later. But first, I would like to take the opportunity and present a few slides about the BioGlue Center, because not everybody is familiar with the center. Competence Center about bio-based adhesives. So this is the situation we have today, this quite urgent situation in the wood adhesives area, where we have, from one hand, a big market volume. The market is quite big, but on the other side, we have adhesives that are based on petroleum resources, and some of them are toxic compounds, so there are health issues about their use. So there's an urgent need to do something. So the BioGlue Center then addresses needs in three sectors: in construction, furniture, and packaging, that all of them use adhesives, in order to help them create commercially viable solutions based on biomaterials, improve health and safety factors, and also facilitate the sustainability profile and recycling profile of products. Of course, we know how adhesives work in general when they bond different materials together, and we know quite well how this works for fossil-based adhesives, but we don't really know how it works for the bio-based adhesives and different rates. And this is what the center is aiming to do, working with fundamentals of adhesion and adhesives, with a vision that I always like to read, that we aim to significantly reduce the dependency on fossil-based materials in the adhesive industry by developing paradigm-shifting knowledge of bio-based adhesives and becoming a world-leading research environment in Sweden. So Sweden produces fossil-based adhesives today, so the infrastructure is there to produce bio-based as well. So to do that, the center has built a consortium along the value chain with three universities, SLU as the coordinator, and I am the director of the center, KTH and Linnaeus University. We have feedstock providers coming from forest and agricultural resources, adhesive manufacturers like Akzo Nobel and BSF, and end product manufacturers in packaging, construction, and furniture, plus associations with Swedish wood. We have grouped projects and concepts in three work packages, and we have tried to identify key research questions in those work packages. Work package 1 is coordinated by KTH and replies the main question: how could renewable materials be used as backbone polymers and cross-linkers for bio-based adhesives? Work package 2 coordinated by SLU, trying to elucidate bonding between bioadhesives and substrates. And finally, work package 3 coordinated by Linnaeus University to expand the application window of bioadhesives in different sectors, and also develop and understand the sustainability gain we get with different bioadhesives. We get funding, and I will explain.
Did we lose Stagios?
I'm sorry for that, but I will just continue telling, because it was just one last slide, that the project is implemented in the form of PhD students and postdocs or other projects. So the duration of the center in the first phase is for 5 years until 2028. And after internal evaluation, we might continue for 5 more years. So ideally, we have 10 years to work in the center, produce knowledge with open access, publications, conference proceedings. And our aim is to create special methods, experts in this area. So in 10 years from now, bio-adhesives will become reality and the center will become as a self-funding entity. Funding comes from three sources. Vinnova is the funding organization, gives 1/3. The industry gives another 1/3 with in-kind and cash contributions, and universities the other third part. So we are aiming for a brilliant future in that area, and we want to tip the scale, and Sweden becomes a world leader in production of bio-adhesives for construction, furniture, and packaging. Again, I'm sorry I was not able to share my screen. And with this small and little introduction, I will now introduce you Peter Herter, who has more than 25 years of experience in product development of adhesives and coatings, with leading positions in research and development. Most importantly, to mention that he has been an R&D manager and then R&D director for Akzo Nobel adhesives for around 27 years. And he has a PhD in physical chemistry for KTH. I used to know Peter since some years now, and we collaborate closely in projects regarding adhesives. And the only thing I can tell is that you can only learn by Peter, and we are very happy and that we have yet another opportunity today to learn something from him. So, Peter, floor is yours now.
Okay, let me see how we do this. So I start. Very ambitious. Thank you for your introductions there, Gus. Very ambitious title, 25 years of adhesives. Let me take you back one step. Why we do this? I mean, my opinion or my view on this is we are facing a dramatic change in the climate. And we need to do something about it and not listen to Trump too much. Adhesives contributes quite a lot to the carbon footprint in the world. So we need to act. And what I like about the whole thing is that when you put restrictions on what you can use in industry, like banning formaldehyde or something like that, you also need to develop new technologies and create a world with new technologies. And just why we are here together now shows that you can drive the environmental agenda in a way that the society gets all the benefits from it. Starting new projects, developing new technologies, and instead of just phasing out and optimizing current fossil-based technologies. So I think the challenge of having an agenda that is based on fossil-free systems creates a world with more opportunities, more jobs, and so on. So it's not the way going back to oil or fossil-based systems. So if we go back to my presentation, it's quite ambitious as it looks here, but I will take you back even further down, 45 years of adhesives. Going back to when I started at the Institute for Surface Chemistry in Stockholm in the beginning of the 80s, I was fascinated by the surface chemical and the world of soap bubbles and lamellar systems, surfactants organizing into aggregates, showing that the world is rather precise at a molecular level. So I got the opportunity to also do my PhD on this topic. So my PhD work was devoted to atomically smooth surfaces, Moscovite mica, glimmer in Swedish. You can actually cleave. This is a mineral that you can cleave into atomically smooth surfaces, which is fascinating as such. And I did work on this and measured what we call hydrophobic surfaces and forces between these surfaces. Imagine that you take these atomically smooth surfaces and bring them closer together. And if you have then a hydrophobic fatty layer on the mica surfaces, you can measure the force interaction between these surfaces with a technique that was developed in Cambridge by a guy called Israel Chvili. And the fascinating thing is that you can go down to molecular distances. If you see the scale on the right-hand side there, these surfaces can see each other, so to say, already at 80 nanometers. That means 800 angstroms. And you can measure the force when you bring them closer together. And if you see the arrow there pointing down, that means that the surfaces jump into close contact. And then you can go the other way around, that you bring the surfaces further apart, and then they jump out into as individuals again, and you can measure the adhesion force that way. So it's a fascinating technique that you can really do science at the molecular level. And so with this as a background, I started to work for Akzo Adhesives, or Casco, it was called at that time, or Akzo Nobel that it's called now. And this is really the basis for adhesion and glues and this field. So I came into the real world of Casco, so to say, working with real adhesives and real systems, and things started to get more complicated, I can tell you. The world that hit me, so to say, at Casco at that time was urea formaldehyde chemistry. The market of adhesives as a whole in the world is quite big, but if you just look at the market for urea formaldehyde glues, it's estimated to 15 million tons per year, and 2030, 19 million tons. So it's growing. The need for adhesives is growing, and that's a general trend, I would say. What we do here with making a resin is that you combine formaldehyde, which is a small, efficient molecule, with urea. And urea is a commodity that has been a very important chemical for the agriculture industry. As you know, maybe the Haber-Bosch method was a successful method developed in the beginning of the last century, where you can make this molecule in an efficient way at very high pressures. And that has saved many lives in the world. So urea is a fantastic product, important. But you have formaldehyde based on methanol, also a commodity. And you get this, what you see on the right-hand side, this resin, urea formaldehyde resin. That is then, when it's used, you add an acid salt or an acid, and you get curing and cross-linking. So the market is huge. And at that time, what was around formaldehyde was health issues. And in the beginning of the 70s, the levels of emissions of formaldehyde was very high. You see in the diagram there, 65 perforator value is a measure of formaldehyde content. And you see it has decreased, and due to legislations and that the market also responds to getting the levels of formaldehyde down. So now we are down to extremely low levels. And I would say that it's not a big issue anymore with formaldehyde emissions from board products. Or the problem could arise in Asia or in the States if you import products that has not been controlled enough, maybe from China and so on. But that's less of a problem now, I would say. But emissions still, and VOC, volatile organic compounds, is still an issue that needs to be highlighted. We spent a lot on this because there was working for industry. There was a threat that formaldehyde would be banned, and that was also a threat to our business at Casco at that time. So we didn't want to stop the articles or so writing bad things about formaldehyde, but we want to have a solid basis, scientific basis for this. And as an example, we talk about formaldehyde, you talk about toxicology. That's what you do in the lab. You can test on animals and so on and see if it's toxic. But you need also epidemiology. That means that you look at people and groups of people having exposed to formaldehyde and see if that you can see any cases of diseases or if people are affected by this. And there is a committee called IARC, the International Agency for Research of Cancer, that we had close contacts with, and they decide the level of how you should brand the product regarding toxicity and so on. And it used to be category 2, but now it's category 1, so it's potentially cancerogenic for humans. But at the same time, it's formaldehyde is part of your cell, the metabolism in your cell. So it's a natural product. So it's just a matter of levels. It's not a matter of if it's in our bodies or not, because the body is used to formaldehyde. But what made the formaldehyde to become more dangerous, so to say, listed dangerous as a carcinogen, human carcinogen, was a study done in the States with 50,000 workers in a city in the US handling formaldehyde. Among 50,000 workers, they found a rare form of cancer called nasopharyngeal cancer. And statistically, you have 6 cases per million of this. And in the study, they found 9 cases. And that difference made the group, the IARC, to come to the conclusion that they need to put a stronger warning on formaldehyde. For us, not being a specialist in the field, it sounds a bit strange, but Going back again, I think formaldehyde, if it's controlled as it is today with particle boards mainly and flooring adhesives, it's not a health issue for humans. But you need to have the control. So the driving forces today is we go from formaldehyde problem, so to say, to LCA. And LCA stands for life cycle assessment. So we look at carbon footprint more than formaldehyde emissions or emissions in general. I like to talk about carbon footprint is a very complicated way of, you get the value and you can't really understand where it comes from. So I like to understand and go back more in the processes and talk about black and green carbons. And black carbons is then fossil-based systems like formaldehyde as it is produced today. And green carbons would then be carbons that comes from what is growing. So I like the approach of green carbons. And the problem is that when you go for green carbons, you might get a higher carbon footprint because the processes are based on black energy, so to say. But that is something you can do. You can do something about that, but you can't do if you have black carbons, they also will still be black, even if carbon footprint will be favorable. So I think the carbon footprint LCA analysis is good, but it's also a figure that could fool you a lot. So you need to be an expert to really understand what you are doing. We made real cases with IKEA actually when I was at Akzo Nobel, and to make it to see really where the emissions come from. We started with production in Kristinehamn, all the raw materials coming in there, and then transportation from Kristinehamn to Hultsfred, where the plant was for making particle board, and you can see where you can make changes. And if you use a truck, of course, then you have the diesel emissions and so on. But you can use train instead, and then you take away that part. So I think you end up in a better discussion if you understand really the processes and how cradle to the grave, how that comes about in realistic examples. So LCA is about understanding the raw materials, manufacturing processes, distribution, use phase, and end of life. Now a little bit about current adhesive technologies. I mentioned formaldehyde-based systems, urea formaldehyde, melamine formaldehyde, phenol formaldehyde, and phenol resorcinol formaldehyde is not so big anymore. It used to be a major part of the business in this field. And then you have non-formaldehyde-based systems, polyvinyl acetate, polyvinyl alcohol, MDI, that's an isocyanate, and starches. The bio-based technologies could be based on starches and could be based on proteins, wood components, lignin, hemicellulose, cellulose, tannins. Lignin is, of course, a very interesting molecule, but it's not very well defined. Lignin depends on where it's growing and how you process it to get the lignin out of the wood, and it varies a lot. And to make lignin more defined, you need to use processes where which is costly, and then you lose the good aspects, so to say, of lignin. Polypeptides is proteins, and they seem to have a natural ability to form bonds and be a binder. Amino acids is, of course, what proteins are built on, and we also have silicates. When you look at bio-based adhesives and challenges, you need to find and discuss the availability of the raw materials. Is it available in large scale, and thus is the purity of the raw material, is it pure enough to get quality of the processes making particle board, for example, that it doesn't vary. And it should be sustainable, of course, and ideally not competing with food resources. You have soy protein, for example, could potentially be competing with food resources, but you also have proteins that are based on for feed for animals, which makes it less of an issue to use. In addition, they must meet industrial requirements regarding quality and process efficiency, and I think this is a step that we need to highlight in all these projects, that we will be able to do research with advanced methods and so on, and that's good. But we also need to connect the results to the real world, so to say. Understanding how do you speed up a resin or the curing of a resin and so on. What is affecting that? So we relate to the real world, and otherwise it won't be. Otherwise, we won't find bio-based glues in the marketplace, and it's not until we get them to the marketplace that there will be a change and impact for the environment. Well, I'll get back to that.
Hello, Peter. I would just like to remind you about the time issue here.
Yeah.
You have a lot to say, but you only have like 15, 16, 17 minutes to go. And I know that you have a lot of slides that you want to show, so you might have to prioritize a little bit.
Yeah. Good.
But please go ahead. Very interesting.
So we have different routes for bio-based technologies. You can do formulations with proteins and starches and so on, and use cross-linkers to make them work. You can use the urea formaldehyde process, and idea that is basically that you can use urea and other aldehydes to come up with new resins. But they use the same mechanism, molecularly the same mechanism. You can do formaldehyde lignin, but then you replace phenol with lignin instead. That's an alternative that is ongoing now also, realistically. And you can go for urea resins based on biogas, where you make formaldehyde from biogas, and then you get sort of 1/3 of the carbon footprint. So this is, you make the same formaldehyde, but you base it on biogas, and then you reduce the carbon footprint of the system, but you use the same chemistry in in principle. So this is a urea aldehyde route. You use glyoxyl, which is a dimer of formaldehyde, another type of formaldehyde that could also be a route. Another way of you can if you use PVACs and isocyanates, which work technically. So if we just make them bio-based instead, that means that you on the PVAC side you need to make vinyl acetate monomers from bio components like ethylene, acetic acid you can make from green carbon, so to say. And isocyanates, the same thing, that you need to base it on aniline from green routes, and formaldehyde from green routes, and phosgene from green routes. And you also get a better carbon footprint. But they are still nasty chemicals, formaldehyde, the isocyanates at least. PVAC is nice in that respect from a toxicology point of view, but isocyanates is nasty. So the toxic aspects comes in here. There are two major mechanisms for adhesives. One is that you form a film. You have, for example, PVACs, you have latex particles they gather together and form a film and act as an adhesive in that way. And then you have the urea resin or resin technologies where you have a cross-link network formed chemically, which is a quite different type of mechanism. And emulsion polymerization is a very nice tool to make PVAC loose. I won't go into that. Now I'm going back a little bit to my thesis. Well, what is a surface? What are atomically smooth surfaces? Some aspects that comes into the bond line is the interaction. Now it becomes a little bit more physical chemistry, which I like, but you have interactions between molecules called dipole, and interactions you have between charged molecules, van der Waals interactions, hydrogen bonds, hydrophilic, hydrophobic effects, and capillary bonds. They all play a role here. And I think you're familiar with the contact angles. You see the droplet on the right-hand side there. You can say that a real surface, atomically smooth surface, you have a contact angle, but with a real surface, with microstructure, roughness, you'll get a higher contact angle. So the real world is more complex than the atomically smooth world. Proteins and sizes. Proteins are in the range of 5 to 10 nanometers. And I like to picture the surface like this. I think if you take the Alps, Austria, the Alps, and say that the surface is like the Alps, then humans would be like protein molecules walking around in the Alps. You take, for example, 100 nanometer roughness of a surface, then you have a small protein molecule there. And what's also acting, of course, is capillary forces. If you have small voids in the substrate, and these are wettable from the adhesive point of view, you get penetration of the liquid and potentially small molecules into these voids, and they play an important role. And we have seen in the project examples that Stagger shown that you can get very nice pictures of penetration into the glue line and so on. So wettability and capillary forces play an important role here. And so does van der Waals forces, as I mentioned before, that the forces between single molecules is, as I point here, very short range. The force is 1 over the radius 6. But if you make the particles bigger, then the forces become much more long range and stronger. So it relates to radius of to 2, R2 instead. So when the molecular structure builds up, forming the resin bond, the forces also increase. The interaction forces increase. PVACs, you see the sizes here in nanometer, 250 nanometer range, proteins. So if you take a real surface here, this is a surface of wood and an example of a glue line. And I don't know if you see the picture. I think it's blocked by Torbjörn's picture, but the half of the picture is 200 nanometers. So you can visualize that you get sort of 10,000 protein molecules into this picture. That's the range of a typical protein molecule. It could also be a PVAC, which is quite bigger than 250 nanometers. But it's good to make these kind of pictures when you try to understand what's going on in the glue line. And for example, if you want to speed up the process, you need to have a faster penetration of the liquid into the system. How do you achieve this? Then you need to have wettability of the capillary systems and so on. So I think it teaches you a lot to understand the sizes and interactions and what could go on at the interface. One major theme in the projects that Stagger's mentioned is cross-linkers, and today we have cross-linkers based on isocyanates, which seem to work together with starches and so on. But we need to develop cross-linkers based on bio-based adhesives too, and bio-based cross-linkers. And this is from a paper by Lydia Heinrich. I think Eva and the team worked with Lydia. Linda worked with Lydia, I'm sure. I think we should go back to that study. I think that was very interesting. And there is also a paper by Lydia that Staggers sent me, and which he can send around to all, which describes potential cross-linkers for bio-based adhesives. But if we can't make it with the bio-based systems, cross-linkers are normally a minor part of the system. So we could still play with the fossil-based cross-linkers to make the whole system work, I think. But in the long run, we need bio-based cross-linkers as well. Finally, here is just a picture of a superglue. Mussels can bond to stones in the sea using polypeptides or proteins, as we call it, and form very strong bonds in water. And that would be the ideal superglue for us. And actually, this is something that they're trying to use and make in large scale, so to say, and sell it as a protein-based glue. But it's very costly and could be used as it is today, only in niche products for certain medical instances. But it's good to think and learn from how does nature do it, gluing in water and get very strong bonds. What's happening here? So some conclusions. I think it's important that we address the knowledge that we build up in the projects, also related to factors that it is of value for the industry. So, and for example, understanding the productivity of industrial processes. If we take Tetra Pak cases with paper-based systems, very fast systems, where you need to understand the watering process, the film forming very quickly, and see how can you make that more efficient. And so also important to understand that the cost per kilo as such is not the major problem. It is the cost per unit that is the key for industry. So it's good to understand that we can still use more advanced raw materials that are costly if they make a good job for the industry. So I think that finalizes the presentation.
Thank you very much, Peter Herder, for your very interesting presentation. And I guess you're trying to not share your screen right now, so we will be able to see each other. And I'm hoping that there is someone who has a question to Peter. We have another 13 minutes for discussion here. So please, we had one question here from Ali Dorie. I'm grateful for your detailed explanation. Anyway, keep in mind the green methanol is becoming available. So formaldehyde might be considered a renewable raw material. I believe bio-based solution combined with TMDI and lignin resins are my recommendations for replacing aminoplastic resins. What do you think, Peter?
Well, many questions there.
Yes.
First, that you have biomethanol, going back to the formaldehyde, the green formaldehyde, so to say. So if you use biomethanol, you can buy it in the market, but it's more expensive, maybe twice the cost or so. And so it's of course a matter of how the bulk situation, how much you produce and so on, if it becomes a bulk product. But what is a bit tricky regarding biomethanol, for example, is that it's based on biomethane, biogas. It has impurities that disturb the process of making formaldehyde, and that's not trivial. So there are costs involved if you go for green formaldehyde, which needs to be addressed.
Peter, can you please turn your video on so we can see your nice face as well when you're speaking? Thank you very much. Is there any other questions in this audience? We have 65 people listening to this webinar, which I think is fantastic, actually.
Yeah.
Mm-hmm. So let's see, is there any hands up in the air or something like that?
It's a bit strange not having an audience when you speak into a.
Well, you definitely have a big audience here, but you can't really see them all.
No.
No. So I got a hand here from Eva, please.
Thank you, Peter, for a very insightful presentation. Really nice. So I think I have two questions. One is again referring back to formaldehyde. I mean, there has been a European wish, or maybe a worldwide wish, to ban formaldehyde. So what are your thoughts about abandoning formaldehyde completely from adhesives? I can start with that question, and then I have a question in regards to recyclability also.
I think it's would be to overdo it. I think you need to have very good control of the emissions because you still see cases where you get products that are not controlled regarding emissions, and that really creates turbulence in the market, and you start to do this kind of ban formaldehyde thinking. But I think the control is with the control there, and there is the great pressure that you need to have control. It's not a good idea to go all the way, because there are other things which is good with formaldehyde. We've seen working with IKEA that you get molds and so on in the particle board using bio-based systems, and then you need to add preservatives, and then what is then the preservative? Yeah, it could be based on formaldehyde because formaldehyde is a good preservative. So the picture is quite complex in a way. It's not trivial, but I think still it's a good drive to go for bio-based adhesives, green carbons, formaldehyde-free systems, because as a whole, if you don't have control of formaldehyde, it's a really bad thing to have. But yeah.
And I think from next year, the values, that allowed values, be even lower than they are allowed now. So I think there is like a strive or an ambition to completely abandon formaldehyde in the long run with the strict chemical regulation.
So I wouldn't put too many resources on going for bioformaldehyde because you will get a slightly better carbon footprint from it, but as a whole, you still have formaldehyde there, which is not a positive name, so to say.
Yeah. So, and then my other question was with regards to recyclability or reuse. So I mean, obviously, we often need to cross-link the adhesives to get the performance that we are striving for. But do you see that is a problem when it comes to recyclability?
You take recyclability as a whole.
Of the board. I mean, of the board. Obviously, you want to keep the wood chips or the wooden pieces in the system for as long as possible.
Yeah, I think that could be a problem. And depending, you need to specify what you are doing. Could it be recycled? Maybe there is one. It could be recycled once, so to say.
Yeah.
Yeah, that needs to be analyzed in detail.
Absolutely.
Based on the specific components you have, I think.
Okay. Thank you very much, Peter.
Thank you for the question, Eva. Do we have any more questions from the audience? We have some comments in the chat that you all can take a look at. My English is not so good, so I'm trying not to read the English chemistry here. But we have from Linda Fogelström a hand in the air.
Yes, thank you very much for the presentation, Peter. Very nice. Could you comment a little bit on sort of the motivation for going over to bio-based with regards to the different types of adhesives we have? I mean, those adhesives that are for applications that are maybe shorter term use and longer term use, and how well they should be. I mean, with regards to mechanical properties and long term use, etc. Could you comment a little bit on that?
I didn't really get your question there, Linda.
But I mean, going over to bio-based, where we should start more or less, should maybe obvious to have those for shorter term use that we produce more of, instead of maybe those that CLT or those that will anyway stand for many years.
Advanced products like for laminated beams.
Yes.
Which needs the best types of adhesives available, which can last for hundreds of years. So there you have quite high demands, and you might think that bio-based systems is not then suited for that. But then you have casein glues, which is a protein, then a milk protein, and that was used to build the railway station in Stockholm in the beginning of the 1900s, 1920, and when they tear it down, the glue line quality was still very, very good. So I think protein chemistry could still make lasting bonds for advanced applications.
That's great to hear.
Thank you very. Yeah, Peter, do you have a last remark, or I'm just about to give the word to Stagios so he can thank you properly and have his last remarks before the time is out here.
No, not really. It's a fascinating world, adhesives, but it's very complex, and I think that going back to my conclusions, that we still need to address the needs in the industry to make this a successful project as a whole. If we do that, it will really pay off for the society.
Great, thank you very much. Stagios.
So, Peter, we are coming to an end here. But before I properly thank you, I just have one last question to summarize also. I know your view about that, but I would really like to ask again. You mentioned different biopolymers as backbones, like starches, proteins, lignins, and there are others like some has comment like glucose, sucrose, and so forth. Do you believe in one or some of them, or you think it can, at the end of the day, you need to combine biopolymers, or even look locally, or adjust to the needs of specific companies for building bio-adhesives?
I think there are many opportunities. I mean, just going for starch, as you have the experience of working with starch, and Resa also at LNU working with starch and modifying starch as such is an interesting component for systems. But I think that the challenge will be for us in the project as a whole to see. We discuss working with model systems, to select model systems that pays off, so to say, that takes a good picture. So I think that will be an interesting topic to discuss, to find, because you can mix protein starches, you can do a lot of formulations and so on, but to build knowledge that is of value for industry, we need to select a few systems to focus on. So I think that will be something for us to discuss.
Yes, that is a very good point. Overall, we thank you very much, Peter. It was a very nice start of those webinar series in the BioGlue Center. I've been very lucky all those years to learn from you, and I'm happy today that others had opportunity to learn something from you. And
thank you.
Let's continue with the good work. It has been a great start. Thanks a lot, Peter.
Thank you.
And thank you everyone attending today, the first webinar. And stay tuned because more will follow and they will be announced at our website by Torbjörn. Thanks a lot, Torbjörn, for moderating. Okay.
Thank you, Sergius, and thank you, everybody. So this concludes the first webinar in a series, I hope. And now I will stop the recording and we will post it on YouTube. So thank you, everybody, for participating here, especially Peter.
Yeah, thank you.
Thank you very much.
Yeah.